our first speaker, Riekelt Houtkooper, who have done similar work in the NAD field, and I'm super excited that he wanted to join. So Riekelt, please come up here, and I will start your presentation. And you can go ahead whenever you're ready.
All right. Is it on? Yes. Thanks, Morten, for the invite, and also to Daniela, Alex, and the whole team for putting up an amazing conference. And thanks to all of you, basically, for sticking around at this time. I think it helped a lot that they have beers and wine to keep you here and preventing you from already heading off to the bar. Speaking of beer, NAD precursors are in beer, so keep that in mind when this talk is passing. So the work that we do in my lab is mainly focused on metabolic networks and how these metabolic networks end up. Oops, sorry, how do I get the pointer? How do you right-click on this thing? All right, perfect. So how these metabolic networks are ultimately leading to metabolic health and healthy aging. And we try to understand how DNA mutations or regulation of expression feed this metabolic network, tune basically how this metro system is working. And we try to understand how nutritional factors or pharmacological agents also impact this metabolic network and in the end promote this healthy aging and healthy metabolism. And one of the pathways that we've been mostly interested in is NAD, nicotinamide adenine dinucleotide. I guess most of you know about NAD, have learned about it in probably your bachelor's or whatever, as a factor that is very much involved in redox reactions. So a lot of the reactions in glycolysis, TCA cycle have some sort of involvement of either NAD plus or, well, probably NAD plus coupled to NADH. So it's a very well-known molecule, plays a role, very diverse role in metabolism. But in recent years also it has become apparent that NAD plus also serves as an obligatory substrate for several families of enzymes, like the PARPs, the poly-ADP ribose polymerases, the sirtuins, and the C-ADP ribose synthetases, with a pleiotropy of metabolic and otherwise functions. So it's a substrate for these, and together these redox functions, as well as these three family of NAD consumers, ensure that NAD has a role in over 500 enzymatic reactions, basically playing a role in everything that is metabolic in your cell. And this also translates to sort of the features of NAD. What does it play a role in? So it's involved in mitochondrial function, redox reactions, as I mentioned. It's involved in circadian regulation, so your day-night rhythm, inflammation, DNA repair, cell division, etc. So a very diverse molecule, and with this in mind also, you can imagine that it's very important to maintain a proper cellular NAD homeostasis. So the question is, how do you maintain this homeostasis? And this relies on both synthesis on one side, synthesis of NAD, and on the other side, consumption of NAD. And roughly translated, that translates to these pathways where NAD synthesis, for instance, is conveyed by tryptophan as an amino acid, so the novo synthesis of NAD, but also recycling through nicotinic acid, niacin, a vitamin B3 analog, nicotinamide riboside as well, also very similar to nicotinic acid, but also the regulation through signaling pathways like AMPK, which activates some of the enzymes that are involved in the resynthesis of NAD. So that's the synthesis side of NAD. At the other side, you have the consumption of NAD, for instance, through CD38, the poly-ADP ribose polymerases, the PARPs, and the sirtuins. And we know that if you inhibit some of these classes of enzymes, like the PARPs, this also leads to a reduced consumption of NAD and thereby increasing the levels of NAD in the cell. So this is sort of how it's regulated. And the fact that this is a tight balance between synthesis and degradation or utilization also is reflected in the kind of diseases that are associated with a disbalanced NAD homeostasis. And I won't go into all the details, but we consider this like a distinction between primary deficiencies of NAD and secondary deficiencies of NAD, where the primary deficiencies are essentially genetic mutations causing in genes that are involved in the synthesis, for instance, of NAD. Several are listed here. And the secondary NAD deficiencies are basically secondary to either a nutritional deficiency like pellagra, which is a very old disease, but has a lot to do with NAD content, but also aging is one of those diseases where NAD decline is at the forefront. And if you want to know more, I just refer you to this paper that we just published. So we've been interested in NAD biology and link to aging for quite some years now. And especially in the days that I was a postdoc in Johan Auerk's lab, we did a lot of preclinical studies in mice and in worms and tested whether NAD was changed with age. And so this is one example. I could have shown several more in other preclinical models. But this shows that NAD is declined with age in young mice. That's, in this case, it's 6 months versus mice that are 2 years old. And I show here the example for the liver, but the same is true for the muscle. And this is associated with an increase of parylation, so PARP activity, as you can see on this parylation Western blot. But the question is, I mean, as I mentioned, we and several other groups have shown in preclinical models that there's this association or this decline in NAD with age. But how about humans? And this is a study that we haven't published yet, that we're currently, we hope to finalize soon. And this is George Janssens, who is in the audience here today, who spearheaded that together with the team of Patrick Schrouwen in Maastricht. And we took muscle biopsies from young individuals and as well from older individuals, older adults. And these older adults were subsetted according to their activity profile. So we had older adults who were, we call them trained. They do 14,000 steps a day, quite considerable. The normal adults, they do around 10,000. I was surprised that they call it normal adults. I think it's quite active. But let's call them normal adults for now. And the third group is impaired adults. So they have clearly a less active lifestyle. We have less of them in this group, only 6. And that's, of course, because if you're an older impaired adult, you typically don't favor a muscle biopsy. So it has been very difficult to recruit these individuals. So we take muscle biopsies from all these people, and then we do targeted metabolomics. We end up with around 140 different polar metabolites that we analyze. And this is basically a description of the data. We see that many of the metabolites that we measure in the muscle biopsies, if you compare young versus old, so this is not subsetted for the activity categories, but just the younger versus the older, is that there's quite a lot of metabolites that are higher abundant in the older individuals, and also a bunch of metabolites that are reduced in the older individuals, and so higher in the younger. And the one that stood out for us, for obvious reasons, is NAD, which is, in this heat map representation, is high in the young individuals and reduced in the older, or I shouldn't say reduced, I should say lower in the older adults. The interesting thing is that if you subset this for the activity profile, so with this, here I call them athletic, so this is the active elderly, the normal elderly, and the impaired older adults, you see that there is also this stepwise lowering of NAD. With the young individuals being high, then if you're an athletic older adult, this sort of is the same level of NAD, but then with the normal older adults and the impaired older adults, actually the NAD is much lower. So this is interesting. I mean, it's a confirmation basically that in a human cohort we can also find these kind of changes that we for years already knew that were happening in animals, and we always assumed that this would be the same in humans. Now we have the confirmation. But the question is, of course, can we fix it? Can we fix this decline and restore NAD homeostasis? And there's many ways to do it, and probably this figure is a little bit too small. Well, here in the room it's okay. There's many different ways to do it. So one is tryptophan as an amino acid is a precursor for NAD. So it feeds into this pathway all the way down here to NAD. And several others are nicotinic acid. So like these are vitamin B3 analogs. And nicotinamide riboside and nicotinamide mononucleotide have received a lot of attention. And I'll show you a couple of examples for nicotinamide riboside because that's the one that we've been working on mostly. But actually, most of what I will tell you also applies to nicotinamide mononucleotide. Several other papers have shown that. So this is a snapshot of some of these results in preclinical models. Nicotinamide riboside basically here prevents high-fat diet-induced obesity and several other parameters like cold intolerance, etc., induced through this metabolic stress. We also found that nicotinamide riboside extends lifespan in C. elegans, so the blue line compared to the red line. Then Johan Auwerk's lab, so my former mentor, showed that nicotinamide riboside also extends lifespan in mice, even when given at a later age. And finally, also his lab showed that nicotinamide riboside, here in orange, delays the proteotoxicity-induced immobility of worms and also improved it in mice. So I think it goes without saying that at least in mice and like these preclinical models, there's extensive evidence to show that NAD increase leads to several health benefits across these different organs and like tissues. But the question is, again, how is it with human situation? Several trials have been published now. I'll give you one example that we were also involved in. Of an NR trial. So this was a crossover design, a placebo-controlled and a 6-week period of treatment. Nicotinamide riboside first and then switched to placebo after a washout or the other way around. And after the treatment period, there was always this sort of intensive like muscle biopsies, clinical phenotyping, and a muscle biopsy was used for biochemical and molecular analyses. So the first thing we tested is NAD actually changed? And we do see, in line with several other clinical trials, that metabolites of NAD, like this one and methylnicotinamide, are actually increased, suggesting that there is an increased flux through this pathway. But if you look at the tissue biopsies, NAD itself doesn't change at all. And we tested several methods of measuring NAD. Still, never we see a change. It is actually in line with the fact that also like this health parameter, like insulin sensitivity, if you look here at glucose excursion and those sort of parameters as a marker for insulin sensitivity, we don't see any changes whatsoever, as you can see in this final column. So treatment with nicotinamide riboside, at least under these conditions, seems to be, well, not too positive. So there seems to be a problem here with a limited translatability from mouse to human. And the question is why? It seems that these classical NAD boosters, like nicotinic acid, nicotinamide, NR, and NMN, they have this limited effect on NAD levels quite consistently. There's some exceptions, like there's a clinical trial from our Finnish colleagues, Anu Suomalainen and Eija Pirinen, where nicotinic acid actually works, but that's a quite extreme set of patients. And the question is, so why does this happen? Is it the duration of the study? Is this 6-week not enough? Is it the dose that we use? Is it not high enough? Is it the focus on relatively healthy subjects? Like I mentioned, patients with mitochondrial myopathy, there it worked. Maybe that's a different situation. And the approach that we took actually is to look at molecules with a more pronounced effect on the NAD pool. And that's the next bit that I will show you. This is actually inspired by Ruben Zapata Perez, a postdoc in my group, who during his PhD developed this molecule. Actually, he's an enzymologist by training. And in his PhD group with Alvaro Sanchez Ferrer in Murcia, they expressed a pyrophosphatase from E. coli and incubated this enzyme with NADH to basically create a molecule that didn't exist before, NMNH, which is basically the reduced equivalent of your regular NMN that we know for some time now. I think this synthesis method of NMNH per se is not very complicated, at least what I understand from him. The complicated bit is actually the purification of the molecule. So he spent a lot of time making this purification work. And even after a couple of weeks, he only has like 100 milligram of the compound. So this is not ready yet for clinical application. But the question was, can we use this reduced nicotinamide mononucleotide, I'll call it from now on NMNH, as a new NAD precursor? And so we first tested in cells. And we used this AML12, a liver cell line. This is a brown adipocyte cell line. This is a human liver cell line, fibroblasts, like human skin fibroblasts. This is a neuroblastoma cell line and the HeLa cells. And in each of these cell lines, we compared side by side nicotinamide mononucleotide and the same dosing of NMNH. And very consistently, maybe with the exception of HepG2, which it's a bit more modest, but all these cell lines have a massive increase of NAD after NMNH incubation and much stronger than the NMN incubation. Then we tested if this is also true in vivo in a mouse model, and the first experiment we did was IP injection with 250 milligram per kilogram of NMN compared to the same dosing of NMNH. And we inject them at T0, so at the start of the experiment. And after 20 hours, we do another sort of a booster injection. And then we take samples, blood samples, throughout this experiment, throughout these 24 hours, several time points. And after 24 hours, we sec the mice, and we take the tissues for analysis. And this is the first data we got. We were super excited, really. At the start of the experiment, NAD is low. Then we start incubate or injecting NMN and NMNH in these mice, and immediately you see an increase of NAD, both in the NMN group, but even more strikingly in the NMNH group. So there's a very massive increase of NAD. But the nice thing even is that there's a sustained increase in NAD after NMNH, because with NMN, even after 4 hours, you already see that NAD goes back to baseline, whereas with NMNH, you see a very prolonged, sustained increase of NAD. Of course, we wanted to look in the tissues, what is going on there. So we tested various tissues, and as with all these NAD-based treatments, it always works best in liver and kidney. So there, again, you see a very massive increase in NAD, especially with NMNH compared to NMN, much more striking. But also tissues that are notoriously difficult to target, like the brain and the heart. With NMN, we hardly ever see increases in NAD. We do see it with NMNH. So that's also quite convenient. So with this, I would like to close. We've seen across our studies that NAD+ declines in tissues across organisms, and we now have also the evidence for humans. We hope to publish that soon. It associates with health status, so the movement patterns of these older individuals, which I think is very interesting, and we need to do further trials to see if this is a causal link or not. We do think that these classical NAD precursors have limited translatability to humans. I think, of course, there's room for more clinical trials, good clinical trials, but we have to see what comes out of that. And I think as an alternative, these reduced NAD precursors may be an attractive opportunity. Though I have to say that we need to do more optimization before we can really apply it clinically. One is scaling up production. That's for sure a big challenge. But also, also the PK, PD, all these kind of tests we really need to do before we can move on. So with that, I would like to close. I would like to thank the people involved. I mentioned them throughout the presentation, but this is the part of the team that really contributed to these studies, especially Ruben and George, but also our collaborators in pathology and the other universities, and of course the funding that made it possible. And I'd be happy to take any questions.
Thank you. Thank you so much. We have a question in the back there.
Yeah, great talk. I had one question with your new NMNH. Did you also look for biological results, whether it increases, sirtuin activity or mitochondrial function or something?
No, that's a good question. We haven't. We have tested the molecule in, for instance, kidney disease models, and there it changes like kidney damage markers, those sort of things. But we haven't checked whether it increases also NAD activity, or like mitochondrial activity, sorry, NAD dependent. Yeah.
Any other questions? We have a question down here.
Very nice talk. I had a question regarding the samples that you took to quantify the NAD levels for the NMN and NMNH. It looked like the first NMN injection had an increase in NADH, but the primer one, the primer injection after 24 hours, it didn't look like it went up like the first injection.
Yeah,
Do you have any thoughts on that?
Yeah, so the second injection is after 20, so I showed you that NMN after 4 hours is actually back to baseline, and the second injection is after 20 hours, and we measure after 24. So that's again 4 hours after the injection. So it probably went up, but then went down again to baseline after 24.
Thank you.
So that's just a timing issue.
Okay, thank you.
I have a question, Riekelt. So do you have any idea about the mechanism? Why is this better at increasing NAD compared with the reduced?
Yeah, that's a great question. And also what we were initially worried about is that it would mainly boost NADH, for instance, because it's a reduced equivalent. But that doesn't seem to happen. So there must be some sort of regulation to push it really more towards the oxidized once it's inside. So the metabolism of NMNH is also a little bit different than for NMN. So it doesn't require NMPT as the enzyme to synthesize NAD. It requires adenosine kinase to synthesize it. So the synthesis route is a little bit different. That was the same for NRH, for instance. But yeah, in the end, we don't really understand. Yeah.
Very cool. Any more questions? Otherwise, let's give Riekelt another round of applause.